Okay, so we started with this glycolysis, the first process of the aerobic respiration. In fact, if you look at the anaerobic respiration, they also start with glycolysis. Okay, so because glycolysis is independent of independent of oxygen. So whether oxygen is there or not, present or absent, glycolysis still take place. Now, don't say that glycolysis only take place in the absent oxygen. Are you clear? Means that when there is a present oxygen, glycolysis won't take place. It's wrong. Okay, be careful. We say that independent of the presence of O2. Are you clear? Whether O2 is there or not, glycolysis will take place. Okay, so the idea here from the word, if you look at glycolysis, glyco here means glucose, lysis means break. So basically we want to split, or we have the lysis of 6 carbon glucose to form 2 molecule of 3 carbon pyruvates. And the entire process here, the idea here is to break down the glucose. Are you clear? To break the glucose into 3 carbon pyruvate. Okay, so the process takes place in cytoplasm and independence of the concentration of O2. So whether O2 zero concentration or high concentration, glycolysis always take place at the cytoplasm. And the entire glycolysis, we can actually divide them into 2 phases here. The first phase we call energy investment phase. Basically means that we invest energy, and then we're going to gain back energy in this energy payoff or energy capture phase. So the idea here is very simple. Okay, try to imagine you want to start a business. First thing you need to invest first, right? You need to buy the product first before you can sell the product. So when you invest, in this case, similar to the glycolysis, before we want to generate ATP, we spend ATP first. It's similar to our business. When we want to start our business, we spend first. We spend for what? Renovation, rent the shop, okay, buy ingredients, correct? So same thing. Before we earn the money, we need to spend the money. Okay. So in this case, we're going to spend how many ATP? We're going to spend 2 ATP first, and then we're going to synthesize back 4 ATP in the energy payoff. So it means that when in this energy investment times, we spend 2 ATP. But at the end of the process, we gain 4 ATP at this energy payoffs. But because we spend 2, what is the net gain? So we say that the net gain here is 2 ATP per glucose molecule. So it means that to break down 1 glucose molecule, we need to spend 2 ATP. By the end of the process, we're going to gain back 4 ATP. So this process takes place to gain this 4 ATP. This 4 ATP is synthesized by substrate link reaction. Sorry, our syllabus is phosphorylation. Are you clear? Through the process called substrate link phosphorylation or substrate level phosphorylations. Okay, so we see that there is a net gain of 2 ATP per glucose molecule. Now, very important to put in the word per glucose molecule, because 1 glucose molecule, we're going to gain 2 ATP, the net gain. Are you clear? If you say 4 glucose, then different story. So that's why you cannot say 2 ATP only, but you have to mention per glucose molecule. Okay, so the process takes place in the cytoplasm. You can see that in this diagram, show you the glycolysis takes place in the cytoplasm. We spend 2 ATP. Okay, therefore, we at the end of the process, we form back 4 ATP. So that is a net gain of 2 ATP. From here, can I see that? Net gain of 2 ATP. Okay, at the process, in the end of the process, you can get another product called reduced NAD. Now, in this diagram, in NADH, in our syllabus, we call it as a reduced NAD. I will tell you guys more later what's mean reduced NAD. And the end of process, we're going to get 2 pyruvate as well. Okay, so products here, we have 3 kind of products. We're going to have net gain of 2 ATP, 2 reduced NAD, and 2 pyruvate when we oxidize 1 glucose molecule. Okay, so now we need to know how to draw this glycolytic pathway, okay? And then we need to know how to describe the pathway as well, okay? Okay, let's look at this glycolysis. So few stages, we don't need to remember every single stage, okay, or steps of the glycolysis, but some steps we need to remember. So first, we always start with the glucose for glycolysis, right? Okay, glucose, which is 6 carbon. Now, glucose is relatively stable. Okay, glucose is relatively stable. So first thing what we need to do here is we make them unstable, make them more reactive. Okay, because activation energy is high. So glucose itself cannot actually break down into 2 molecules of the pyruvate. Are you clear? We cannot lyse, I mean break down, right? We cannot break it. Okay, so in this glycolysis process, we need to actually spend the ATP first. So for 1 molecule of glucose, we need now 1 molecule of ATP to phosphorylate the glucose. Okay, can you see that ATP donates the phosphate group? So when ATP donate the phosphate group with the glucose, so the process is called phosphorylations. Are you clear? But it's not substrate level phosphorylation because ATP is not a substrate here. ATP donate to the substrate, so different story. Okay, so ATP now become ADP. So okay, sorry. So 1 molecule ATP, 1 molecule ADP. So it means that now glucose become glucose 6-phosphate. Okay, also 6 carbon, 1 molecule. Are you clear? Clear not so far? Okay, so it means that the first process we have done. So why want to do this? So the answer question also we always ask why we want to phosphorylate the glucose molecule. Why you want to phosphorylate? Because when you phosphorylate the glucose molecule, so glucose molecule actually gains the ATP. Okay, so gains the phosphate group, so therefore they become reactive. Then this glucose 6-phosphate will be converted into fructose 6-phosphate. Okay, 6 carbon. So in some books, actually they merge both steps together. They skip this step, but never mind. We just learn in full. Now, fructose 6-phosphate not enough. Energy is not enough to break them yet. So therefore, fructose 6-phosphate will gain the energy level, increase the energy level. How? ATP. So we spend 2 molecule ATP. Okay, so therefore now become fructose 1,6-bisphosphate. Bis mean 2. Okay, 6 carbon. So can you see that again, guys? So can you see that we spend 2 molecules of ATP? Can you see that? We spend 2 molecules of ATP now. Okay, so from glucose, we gain 1 phosphate group from ATP. After that, fructose 6-phosphate also gains. Okay, now the 6 here and 1,6 means a location at carbon 1, carbon 6. So it means that the phosphate group now attached to the carbon 1 and carbon 6. So now they have high energy level already. So what will happen? So lysis take place, splitting lysis take place. So from 1 molecule of this fructose 1,6-bisphosphate, we're going to get triose phosphate. Now triose phosphate is 3 carbons, correct? So it means that we're going to have 2 molecules. So for 1 molecule of glucose now split into 2 molecules of triose phosphate. Are you clear? So the one I circle basically means that's the number of molecule. So from this part now, we know that we have done for the energy investment already. So we spend we spend ATP. Can you see that? So we have done for the energy investment phase. Okay. Now, clear or not under this part? You need to remember, the step. So glucose accept one phosphate group from the ATP. So this process is known as phosphorylation. Okay, this also, so two-type phosphorylation. Okay, and then this one we call as lysis. Okay, so we lyse, okay, or we break the fructose 1,6 bisphosphate into 2 molecule triose phosphate. So now we spend the ATP already, we want to gain back ATP. So how we gain back ATP? So this triose phosphate will undergo a series of reactions. Okay, so this series of reactions is termed as the oxidation to become pyruvates. Are you clear? Okay, so from these triose phosphates, you go through a series of reactions, then we're going to get pyruvate. Okay, so during this process, what actually happened? Okay, so first, we have the substrate level phosphorylation. So it means that ADP now become ATP 1A. So because 2 molecular triose phosphate, so with 2 and 2. So this process is by substrate level or substrate link phosphorylation. Okay, and then we need to oxidize. Okay, we need to oxidize the triose phosphate. We always say oxidize the glucose. Actually, oxidation only take place now. So what's been oxidation? Now understand the concept of oxidation before we continue. So if we look at oxidation, oxidations either lose the electron, cannot. Okay, so release the electrons. Okay, can you see that? Or we're going to lose hydrogen atom or gain oxygen atom. Cannot win oxidation. So reductions. Reduction here, actually, we see that we gain electrons, so reductions, or gain hydrogen atom, or lose the oxygen atom. Understand the concept here. Okay, so in this process, what will happen here is the triose phosphate undergo the series of reactions, okay, to form the what we call the intermediate compound. Now, in our syllabus, you do not need to know what is this intermediate compound. You just see the intermediate compound enough already. So this intermediate compound also 3 carbons, 2 molecule. So this 2 molecule intermediate compound going to release the hydrogen atom. So they release the hydrogen atom. This hydrogen atom will be picked up by who? This hydrogen atom is released. So this hydrogen atom released, and therefore we have the NAD. NAD is something like oxidizing agent, oxidize the intermediate compound now. Okay, so because NAD going to gain the hydrogen atom, so NAD now become reduced NAD. So 2 molecule NAD, 2 molecule reduced NAD. So now you oxidize the compound, intermediate compound to another intermediate compound. Which is 3 carbon as well. Okay, so what actually happened here? What should happen? So look at this. NAD going to gain the hydrogen atom to form reduced NAD. So this is actually a process of oxidation and reductions. Can you see that? Oxidation reduction. Why we say this? Because intermediate compound is oxidized. Okay, NAD to reduce NAD actually reduction. So it's a redox reactions. Can you see that? Okay, oxidation when? When is oxidation? When an intermediate compound is oxidized. Okay, where is reduction? When NAD now reduced to reduce NAD. So reduction take place onto the NAD because NAD is an oxidizing agent. Okay, and the last part here, substrate level phosphorylation take place again. Okay, ADP become ATP. So 2 molecule. Okay, so again, substrate link or level substrate link phosphorylation. So we have done until this part. So from here, from triose phosphate all the way here, we call energy payoff. Mean that we gain back the N, right? The ATP. Okay. So this I go through again with you guys. So wait, get ready my pointer. Okay, so you can see that first we have glucose, and glucose is relatively non-reactive. Okay, so they won't split into the pyruvates immediately. Okay, so let me enlarge it a bit. You can see it. Okay, can you see that? Okay, glucose. So therefore, we make them have higher energy levels so that they can overcome the activation energy of the process. So therefore, we use the ATP to phosphorate glucose. When glucose receive one phosphate group, so it becomes glucose 6-phosphate, glucose 6-phosphate. And then we have the conversions here, convert the glucose 6-phosphate, become fructose 6-phosphate, and fructose 6-phosphate now gain one molecule of phosphates. Are you clear? Can you see that? Again, the phosphate group. So therefore, fructose now from one phosphate group, we have 2, 1 and 6 attached to 1 and carbon 6. And now they have sufficient energy. Lysis process take place. We have 2 from one molecule of fructose 1,6-bisphosphate. We get 2 molecule of triose phosphate, which is 3 carbon. From 6 carbon become 3 carbon. So this triose phosphate, generally we say that is oxidized, because the whole process oxidation cannot. So we say triose phosphate is oxidized into pyruvate in a series of reactions where 2 ATP, okay, will be synthesized. Can you see that? 2 and 2. But because we have 4 and we spend 2, cannot 4 we earn, we spend 2. So therefore, that is a net gains of 2 ATP per glucose molecule. Okay. Clear or not, guys, so far? So what are the products formed in this glycolysis? Okay, so per glucose molecule, one glucose molecule, per glucose molecule. So you can see that we're going to have at the end, what's the end product? What's the end product? Entire process take place under the end, we get 2 molecule of pyruvate, 3 carbon. So we get, okay, a net gain of 2 ATP. So basically we synthesize 4, we minus 2. So therefore we net gain of 2. And last we have 2 molecule of reduced NAD. And reduced NAD is very important in oxidative phosphorylation. So this one later will be used in oxidative phosphorylation. Okay, clear? Any more questions so far, guys? So during this energy investment phase, to make 6 carbon more chemically reactive, glucose is phosphorated by ATP, forming 6 carbon fructose 6-phosphate. So means that in this case, I skip the glucose 6-phosphate. Okay, so it actually it starts with your glucose 6-phosphate, convert to fructose 6-phosphate. And then this fructose 6-phosphate receive another phosphate group from ATP, forming fructose 1,6-bisphosphate. And we know that now they are very, very reactive. Okay, it can cleave by the enzyme to form 2 molecule of 3 carbon triose phosphate. So in this process, energy investment phase is completed. Okay, so now we begin with the payoff phase. So this 3 carbon pyruvate, okay, or 3 carbon triose, 3 carbon triose phosphate, we oxidize them to form 3 carbon pyruvate. And during this process, NAD is reduced to reduce NAD. So this basically you can write down, NAD actually receive not proton, but hydrogen atom. Be careful when you write, it's a hydrogen atom. So when you receive hydrogen atom, it itself now become reduced NAD. And a total of 4 molecule ATP actually is formed by substrate level phosphorylation or substrate link phosphorylation, okay, forming 4 molecules of ATP. Okay, but what's the net gain? The net gain only 2 because we spend 2. Clear? So in these diagrams, actually, you do not need to know. In fact, you can see that we have whole full process. Actually, okay, it's not so simple. But again, as I say that you do not need to know everything. Okay, the structure. So you can see that glucose is alpha glucose. Can you see that? Okay, so accept one phosphate group. Can you see that? Accept one phosphate group at carbon number 6. Okay, and then accept another phosphate group linked at the carbon number 1. Okay, so that's why it's a fructose 1,6-bisphosphate. Okay, at carbon number 1 there. So then splitting process takes place. In fact, the splitting process is not equal. You can see that we form one molecule. Can you see different, right? Can you see that? One is a glycerylrehyde 3-phosphate, another one is dihydrogenone, right? Phosphate. But again, because interconversions, then glycerate 3-phosphate will continue the process. So during this process, you can see that we can form the NAD and then we gain back the ATP. Can you see that? Okay. So in this case, you will gain back the ATP. Can you see that? We gain back ATP. So this is from one glycerylrehyde. So means that we have another one. So basically 2. Okay, so total we have 4 molecules. Okay, so again, do not need to worry about this diagram. It's just the overall process, overall process here. Okay. Now, what are the products of glycolysis? What are the products? So per glucose molecule, we have net gain of 2 ATP, and then 2 molecules of reduced NAD, and 2 molecules of pyruvate, which is 3 carbons. Okay, so we have done for glycolysis.